Hey class, in this video we're going to focus on cell division. We're going to focus on stages of the cell cycle, and I want to really just emphasize here, we're going to be kind of sticklers, we're going to be kind of pedantic about mitosis versus cytokinesis. So as we think of mitosis, I want you to fixate on division of the nucleus. Cytokinesis is division of the cytoplasm. And I know at the very top of your lab objective sheet it says, utilize all histology links for all objectives marked with an asterisk. Well, this is the first time this semester or in this class we're going to see these asterisks. So you need to be able to recognize what these things are on a photomicrograph. So to give you an idea, you can check your lab manual. Figure 4.4 in your lab manual does a great job of that. If you load the A&P app in Visible Body, it's sections Chapter 2, Section 8, 12, and Chapter 3, Sections 2 through 4. I'm a big fan of digital histology, so I'm loading that up right now for us here. And here we can see interface. And as we look at this digital histology site, it's fantastic. We can alternate between unlabeled and labeled slides. I also want to emphasize here that this digital histology website includes a lot of extra information. So for instance, cell membranes, they're good to know what those are, but if we look at our lab objective sheet, we don't have any cell membranes on the lab objective sheets. So I want you to not focus on those extra things. We're going to skip ahead to some whitefish blastula cells. So I'm going to go ahead and maybe make our screen a wee bit smaller here. Here's some whitefish blastula, and we have the different stages. And when we focus on prophase, I like to focus on late to mid prophase. So as we're looking at this stage, prophase here, the first part of mitosis, we are going to have the chromosomes condensing out. So it kind of is a heterogeneous looking. It's not, instead of being homogeneous, it's heterogeneous. And that heterogeneous appearance of the nucleus is the chromosomes condensing out of the chromatin. So if you see purple speckles where the nucleus is, there's your sign. We're looking at prophase. And then as we continue on, after prophase, we're going to have metaphase. So we're just going to go ahead and skip that slide, because we want to focus on the whitefish blastula slides. Here's our next slide here. For metaphase, I want you to think metaphase for middle. Middle. During metaphase, right here, we're going to have a metaphasic plate. This metaphasic plate is when all of the chromosomes are lined up right in the middle of our cell. As we look at our lab objective sheet, we have metaphase and metaphase plate as two of our objectives. On our next slide, we're going to have anaphase, prophase, metaphase, anaphase. So let's pull up anaphase here. When we look at anaphase, I want you to fixate on chromosomes that are being physically ripped apart and pulled to opposite ends of the cell. So after the chromosomes line up in the middle during metaphase, they are going to be ripped apart and pulled to opposite poles of the cell. I kind of like this mid anaphase right here. I think this is a good money shot right here. And as we're looking at this, if I can zoom in on us, oh, there we go. We can see how we have the sister chromatids on the left and the right that have been separated from each other. That's pretty cool. After anaphase, we have telophase that we need to be able to recognize via a photomicrograph. So let's go ahead and skip ahead to telophase. And telophase is one of those things. Oh, here we go. We are going to skip that. We're no electron microscopy at all. I want to emphasize that telophase overlaps with cytokinesis. So telophase of mitosis overlaps with cytokinesis. So telophase is when we're just starting to form two brand new nuclei. So you can see we have 1, 2. Let's go and hit late telophase here. We have 2 new nuclei forming. If we look at early telophase, this is probably what I fixate on the most when I'm looking at physical microscope slides in the lab. We have 1, 2 brand new nuclei forming. And that's going to be the telophase. So what stage of mitosis is this? It is telophase. And now if I was to ask you what this indentation right here, let's go ahead and annotate that screen for us. Boom, boom, right here. We can see how there's kind of an indentation. That indentation that goes across right there, that's referred to as a cleavage furrow. And that cleavage furrow is when the cell membrane starts to pinch off and form two brand new cells. So this is the separation of the cytoplasm, or division of our cytoplasm. I'm going to go ahead and close that up here. Boom, boom. Come on. Come on, I want that menu to go away. Go away menu. Ay, ay, ay. Well, there, that works. So that was cytokinesis. That's the division of the cytoplasm, and that indentation was the cleavage furrow. So, class, that was all we have for our discussion of exercise number 4, stages of the cell cycle. If you have any questions or comments for me about this material, please don't hesitate to post them on the class discussion board or to shoot me an email. And as always, happy studies.